listeners, welcome to episode 105 of Cell and Gene, the podcast. I'm your host, Erin Harris, and today's episode is all about regenerative medicine, specifically the therapeutic potential of extracellular vesicles, or EVs. I had the pleasure of sitting down with Dr. Gloria Matthews, Chief Medical Officer at Aegle Therapeutics, which is a clinical stage company developing regenerative therapies for rare and severe dermatologic and immunologic disorders. Aegle is pioneering the use of EVs derived from mesenchymal stem cells to restore damaged tissues and not just heal them, but regenerate them. Dr. Matthews shares how these tiny bioactive vesicles may offer powerful advantages over traditional cell-based therapies, especially in treating debilitating diseases like recessive dystrophic epidermolysis bullosa, or RDEB, often referred to as the worst disease you've never heard of, which she gets into during our discussion. And so from their promising clinical trial data to the challenges of scaling biologic-based treatments, my conversation with Gloria is packed with insights into what might just be the next evolution in regenerative medicine. And so without further ado, here is my conversation with Aegle Therapeutics, Dr. Gloria Matthews.
Hey, Gloria, thanks for being here with me on Cell and Gene, the podcast. Happy to have you.
Thank you. It's my pleasure, Erin. Thanks for having me.
Good. Okay, so talk to Cell and Gene, the podcast listeners, about Aegle Therapeutics and its mission.
Well, Aegle is a clinical stage regenerative medicine company. We work to develop therapies for rare and severe dermatologic and immunologic disorders. We work to extend the lives and improve the quality of life for patients suffering from these devastating diseases by restoring affected tissues to a more normal state. So we focus on not just healing, but regeneration, and restoring back to the way tissues were meant to be. We provide treatments that are effective with added safety without unintended and sometimes intolerable side effects, such as severe itch or exacerbation of that disease.
Excellent. Okay, thanks. So Aegle Therapeutics is known to be pioneering the use of extracellular vesicles, or EVs, derived from mesenchymal stem cells. Can you explain how EVs function therapeutically and what advantages they may offer over cell-based therapies?
Sure. Well, we know that MSCs are known to be crucial for tissue repair and regeneration. We know they provide bioactive signals that modulate immune response and inflammation and drive regeneration, which are our goals. This can lead to effective healing. The bioactive cargo is that they deliver sequestered in these little tiny carriers or extracellular vesicles that are waiting to be secreted from the cell. And literally billions of these extracellular vesicles can be secreted from these cells. Then they are capable of delivering cargo such as genetic material, DNA, RNA, proteins, signaling factors that can reduce inflammation, modulate the immune system, and even provide regenerative healing cues. In fact, we now know that this is the way that MSCs exert their effect on local cells in the body. Unfortunately, the MSCs themselves are rapidly cleared from the area in which they are injected. This often results in lower efficacy than we might expect from laboratory studies and even from some clinical studies that have been conducted. Some of the main advantages of EVs over cells are that the EVs that are injected or applied locally fuse with the cells and the tissues and deposit the bioactive cargo into the cell. So they provide the benefits of the MSCs without the concerns around residence time of the cells or survivability of the cells in the body, which tend to be rapidly cleared once they're administered. The cells, that is, not the EVs. EVs are not living cells. They're easier to create into a product format and to maintain long term. EVs can make billions of these, and there is a limit to how many MSCs one can culture from an individual donor, but their MSCs, a set of MSCs make EVs that then can amplify that ability to respond exponentially. We can do precise dosing. This can be delivered in terms of numbers of EVs in a given dose volume without concerns about cell death or proliferation or any kind of differentiation of the cells. They're very robust in harsh environments. They don't engraft in the tissues. They are easily destroyed. So they're really provide an off-the-shelf option for people. And I guess the last thing I'd say is that, there are only so many MSCs that can be harvested from an individual donor. And as I mentioned before, each cell makes, volumes of EVs. So these can be purified and result in an efficient scale up with less concern regarding the eventual availability of the product and donors.
Nice, nice. Okay. I want to talk a little bit about your lead product, AGLE 102. And as I understand it, it's being developed for severe dermatologic and inflammatory conditions such as DEB. So, talk to us a little bit about, first of all, what is DEB and what have been the key insights or findings from your clinical work so far in in the rare and complex diseases?
Sure. Well, DEB or epidermolysis bullosa, EB, has been referred to as the worst disease you'd never heard of. So lots of people, even in the dermatologic medical community, have either never seen or never heard of EB. It's really a rare disease. There are multiple forms. We are looking at recessive dystrophic epidermolysis bullosa. There are another few forms that I won't go into in the interest of time. But these are severe skin blistering diseases, and especially dystrophic and recessive dystrophic. These are very severe diseases, and they're caused by a genetically driven inability to make collagen protein or make effective collagen 7 protein. And they also exhibit chronic inflammation and immune imbalance. So it's not just about collagen 7 deficit, which would be easier to just replace as in a protein replacement therapy. But we also have this chronic inflammation and immune imbalance. So the collagen 7 is critical for anchoring the epidermis to the dermis. And when you can't anchor those two in its absence, which is particularly severe in the recessive form, which we call RDEB, recessive dystrophic epidermolysis bullosa, this results in lifelong formation of recurring blistering wounds all over the body, with significant healing delays, significant scarring. These wounds often lead to early and recurrent development of squamous cell carcinoma, which in many cases can be fatal even at a pretty early age. Clinically, some of the things that we've seen in our current ongoing phase 1/2a clinical trial is that, first of all, they're remarkably safe, even in severely compromised patients. We've also done clinical studies in deep second-degree burn wounds where people, presumably healthy people, are suddenly compromised by significant burns from gasoline fires or other pretty damaging explosions. And these have been incredibly safe. They rapidly stimulate bioactivity in chronic non-healing wounds that is not seen in matched control wounds. In our RDEB trial, we currently provide up to 6 doses 2 weeks apart in those patients, and we see detectable changes start as early as after the first dose. So we check them 2 weeks later, and we start to see differences in the bioactivity of the wound. And sometimes it takes a few doses to close. Sometimes they close very quickly, and sometimes they don't. But even before full wound closure, we see the patient's symptoms and quality of life improve based on both patient and physician-reported validated outcome surveys. So it's been quite encouraging. The wound closure we see is also notably regenerative, with restoration of the dermal layers. And that's something that if you're just supplying collagen 7, you're helping with the anchoring of the epidermis to the dermis, but there's really no opportunity to provide dermal healing with just supplying col 7. That's really just kind of a restoration of the bungee cords, if you will, connecting the dermis and the epidermis. Our preclinical work in the immunologic disease graft-versus-host, in which a recipient of, say, a blood cell transplant is immunologically attacked by those cells, by the donor-derived cells, which see the recipient as a threat, has also shown remarkable reversal or prevention of these effects using systemic administration of EVs, which is departure from our local administration of the wounds. But it does suggest significant potential beyond the topical application.
Gotcha. Thank you. Okay. One of the most exciting aspects of EV-based therapeutics is the potential for off-the-shelf treatment. Talk to us a little bit about what you think the biggest hurdles are facing the development of EVs, into scalable, commercially viable products.
Sure. Well, Erin, these are biological products, right? Derived from human donors. And just like cell therapies, there's not a perfectly neat fit into the regulatory infrastructure with the FDA and other regulatory agencies. That's just a fact. The challenge has been mitigated over the years as regulatory authorities have seen more of these, gain more exposure with, experience with these types of therapies. There is a concern with the recent changes in the US FDA in terms of leadership, reviewers, staffing, and project management, that some of the institutional knowledge that the cell and gene therapy industry has worked to build over time could be lost. So that's one of the challenges that we're concerned about. Also, the capabilities of the FDA to accept submissions and meet their established review deadlines is likely to be compromised. So we're keeping a close eye on that because this was reflected in longer development times for companies. These development times are already long. That's just the nature of the beast. This can often be sustained by larger pharma companies, but small biotechs and biopharmas are at risk because they can't maintain their funding for these longer development timelines. So those are really very contemporary challenges. There's also more practical challenges at the level of manufacturing. Scaling up manufacture at commercial scale in the GMP environment have always been challenging for cell and gene-based therapies and other cell-based therapies. As we contend with donor-to-donor variability, an establishment of robust master cell banks to supply the product, minimization of losses in product yield due to necessary filtration and purification processes. And frankly, experience in industry has demonstrated these challenges are surmountable. Quite a few entities have developed these types of products through GMP manufacture at scale. In some ways, the cells in this case are the sort of weak link, particularly MSCs in the manufacturing chain for EVs. The MSCs kind of a limited proliferation capacity. We don't take them out very far in passage. There's evidence that as you passage them, they start to lose some of their regenerative capacity as you get out in the longer out in the passages. But they produce billions of EVs, right? So once they're purified, tested, packaged, they're stable and they can be maintained as an off-the-shelf product for extended periods. So making banking of commercial supply of EVs entirely feasible.
Good. Thank you. Now, as we talked about, as you mentioned earlier, certainly, inflammatory skin and tissue disorders are kind of notoriously difficult to treat. So from your perspective as a chief medical officer, what makes EV-based approaches particularly promising?
EVs provide a multifaceted approach. So they're non-immunogenic like their parent cells. They modulate rather than exacerbate inflammation and immune dysregulation. They're suitable for both local and systemic delivery. We've shown that both in our preclinical models and in our clinical settings. And for dystrophic EB, you can deliver missing genes and proteins such as collagen 7, but beyond just delivery of collagen 7, you also have the ability to treat both dermis and epidermis by delivering proteins and nucleic acids that are needed to build the anchoring fibrils and promote scar revision in the dermis while regenerating tissue and also reducing inflammation. So it's really a multimodal approach. Resupplying collagen 7 alone, as many of these sort of first generation, if you will, products have been able to do. We have some gene therapies and other transduction therapies in autologous skin grafting that have been recently approved, and they resupply collagen 7, and they're an important part of the therapeutic regimen. They can promote epithelialization of the dermal layer by providing, of the epidermal layer, by providing collagen 7, but they can't necessarily promote regenerative healing of the dermis. So we really see this as kind of Gen 2, if you will, of the toolbox of treatments that can be done for dystrophic EB.
Great. Okay. So beyond dermatology, what other therapeutic areas or indications do you think are well suited for EV-based therapies? And are any of them on Aegle's horizon?
Sure. As mentioned, there's great potential in the treatment of graft-versus-host disease. They are also potentially suitable for any indication that has an immune inflammatory component, particularly those for which local delivery is desirable, such as ocular and orthopedic disorders. Also, I know you meant to move away from dermatology, but there's also the potential for expansion to other EV subtypes beyond RDEB, as well as other severe derm indications. So there is quite a bit of potential there for this type of therapeutic.
Excellent. Love that. Okay. Before we wrap up, what is next for Aegle Therapeutics? What's on the roadmap?
Sure. We currently have fast track and rare pediatric disease designations granted by the FDA. We're at a stage where we have multiple GMP manufacturing runs produced for adequate supply for multiple clinical trials. We have finished collecting data on a deep second-degree burn study and have 65 branded patents, currently completing enrollment of the RDEB phase 1/2A study. So all these things we feel put us in a good spot to be moving into pivotal studies that will get us to a marketing application such that we'll be able to commercialize EVs for RDEB. In parallel, we are also raising a new series of funding to enable additional discovery and some preclinical work to advance our programs in other rare disease indications, including other forms of EV and other immunodermatologic indications such as hidradenitis suppurativa or HS. And as I said, we're pretty far along in our preclinical evaluation of graft-versus-host disease.
Excellent, excellent. Well, thank you so much. And so that's a wrap for this episode featuring my guest, Gloria Matthews of Aegle Therapeutics. Gloria, thank you so much for this time and all the great insight you shared with us today. This was wonderful.
My pleasure, Erin. Thank you for the opportunity.
Absolutely. Please visit us at cellandgene.com and be sure to subscribe to Cell and Gene, the podcast, so you never miss an episode. And we'll talk to you soon.